Australia is like a unique place. We have so many endemic species, which means we have species which we can only find in Australia, or maybe even only in certain regions in Australia. We really need to protect this because really we rely on living together with nature.
Plants, when you look at them through a garden, they might be covered in various spots and dots, but is that actually a spot and dot of concern? In the case of myrtle rust, it's quite a devastating pathogen.
So myrtle rust is a very devastating fungus because it infects over 300 of our native species, ones we really love such as eucalypts. Really our native species don't have resistance genes against myrtle rust, and this has really led to 5 of our native species becoming critically endangered because of myrtle rust.
So we have so many native species that are on the brink of extinction from myrtle rust, like this Melaleuca, and it's really important to understand the disease better to protect our natives.
So we're going to have a very unique network of samplers all the way around Australia's coastline, in Australia's major botanic gardens, undertaking the same sampling strategy and the same approach of trying to understand what's going on with myrtle rust.
We're able to find where have we got these fungi, where are they prevalent, and how much.
And part of our work is to understand how we can breed resistant plants to myrtle rust.
It's definitely a driving motivator to contribute to caring for country so we can pass it on to future generation, to our own kids, grandkids. Spores are all around us, fungi are all around us. Air we breathe is full of particles. It might be pollen. Often it's also like fungal spores. So fungi release these really tiny particles which you can only see with the microscope.
A spore is the way that fungi try to reproduce, and so they put them out into the air hoping that it will land on other fungi.
And then also, of course, they can land on a new leaf, and when they land on a new leaf from another plant, then they can infect this new plant.
Here we're looking at a heavily infected leaf of a Myrtaceae plant, and you can see the very characteristic yellow spores of myrtle rust.
Plant pathogens are designed to move their airborne spores through the air, and sometimes short distances, but sometimes great distances as well.
So we've put in an air sampling device here at the Botanic Gardens that is able to trap fungal spores from the air.
It's like a big hoover where you collect the spores that get condensed down, and we can then analyze them afterwards.
We can then get the samples from the air sampler and we can DNA sequence them. And then by looking at the DNA sequence, we're able to identify exactly what is in the air.
This is the intake where the airborne spores and airborne biota are sampled within the air column above me. Those particles are sampled into a specific little barcoded vial, similar to the one that you can see here.
Once we collect the spores from the spore trackers at the Botanic Gardens, we do a DNA extraction, and then we prepare a DNA library, and we're at this stage now where we actually sequence that DNA to determine whether there was myrtle rust in those spore tracker samples we collected or not.
So we had this proof of concept experiment first here with a larger machine, which is basically the back of a truck, much bigger, much more complicated and so on. And then really scaled this up to innovate both in the spore samplers, which come from the back of the truck, to really a suitcase, and also the diagnostics at the back end. Lots of innovation went there, and also it allowed us then to go from 1 botanical garden or 2 botanical gardens to really 9 botanical gardens, to make it like a whole Australia-wide initiative. How we optimize these reactions, make them cheaper, and also scale them from 50 to hundreds of samples to thousands of samples to be analyzed.
So this is a really amazing amount of data that's been generated for the first time in Australia, where we can actually see the load of different fungal spores that are being spread around and really correlate that to real-time data.
This becomes a really valuable information stream that we can give them the heads up that something might be occurring, and we are detecting it in the air, and can you now have another look?